recording for this session. So in the previous lecture, we were explaining the anatomical structures in the upper respiratory tract and in the lower respiratory tract. And if you still remember, the upper respiratory tract consists of the nose, the nasopharynx, oropharynx, laryngopharynx, and the larynx, trachea, primary bronchi, secondary bronchi, tertiary. Okay, sorry, the upper was until the trachea, and then from the trachea down to the bronchi, that's considered as the lower respiratory tract. And then we had another classification of the conducting zone and the respiratory zone. The conducting zone was starting from the nasopharynx down into the respiratory bronchioles, and then from the respiratory bronchioles to the alveoli. Those areas where gas exchange happen are considered the respiratory zone. So today we will be discussing the respiratory system physiology, and as you know, in the physiology we need to understand how it works and how ventilation happens. So the learning outcome for today's lecture is to understand the process of ventilation, the mechanisms, the pathways, the muscles which are involved, the pressures, which are three types: alveolar, pleural, and transpulmonary pressures. We'll understand the respiratory center and the different types of groups of neurons inside it. And then we will proceed into the factors that could regulate the breathing mechanisms. And then we'll understand the concept of lung compliance and surfactant, respiratory distress. We will go very fast through the lung volumes and capacities, dead space, which we have already explained before, and then the important topic of gas exchange for oxygen and carbon dioxide. And we will go into hypoxia, hypercapnia, illustrate number of causes which can lead to these two conditions. And at the end, there will be a number of common respiratory problems that will be explained. So can you hear me clear? Everything is okay? Okay, good. All right, I will proceed now. So we will start with the process of ventilation. That process aims to deliver oxygen and to wash the carbon dioxide from the cells. And this process has many stages. It starts with the first stage, known as the pulmonary ventilation, whereby there is an inhalation and exhalation. Inhalation, also known as inspiration. Exhalation is also known as expiration. Here, the air will flow from the atmosphere, which is the environment surrounding us, into the lungs. And this process of movement in and out is ventilation or pulmonary ventilation. That's the first step. This will be followed by external respiration. So external respiration is the gas exchange that happens in the respiratory membrane between the air in the alveoli of the lung and the pulmonary capillary circulation, which is part of the respiratory membrane. And in this process, oxygen will be delivered to the pulmonary circulation. Carbon dioxide will be released into the alveolar airspace. And this is the external respiration. After that, of course, there is one step, which is the blood circulation, transferring the oxygenated blood from the pulmonary circulation back to the left side of the heart, first into the left atrium, then to the left ventricle. Then from the left ventricle, the circulation will, the blood will be distributed into the systemic circulation. And once the blood reaches into the capillaries at tissue level, then another level of internal respiration will happen of gas exchange between the capillaries and the tissue cells. And in the last step, when the oxygen is utilized inside the cell for metabolic reactions such as Krebs cycle, aerobic glycolysis, whereby ATP is produced, that is known as cellular respiration, when the oxygen is utilized inside the cell. So these are the four different stages of the ventilation process. So I will focus mainly, because today's lecture about the physiology of the respiratory system, so we will be mainly focusing about the first two, the pulmonary ventilation and the external respiration, with the gas exchange. And the other parts are going to be discussed when the system is going to be within that topic. So we start with the pulmonary ventilation. In pulmonary ventilation, as we mentioned, that it consists of two processes: the inspiration, which is the movement of air from the atmosphere into the lung, and the expiration, which is the opposite, movement of air from the intra-alveolar space, from the intrathoracic cavity into the atmosphere. So how does the lung move from in these conditions? How does it move in and out from the lung? Well, this follows a very simple mechanism, which is related to the pressure of air. So air moves simply from areas of high pressure to areas with low pressure. And that's how the lung can take air in or push it out, by changing the pressure inside the lung. As we all know that the atmospheric pressure around us is constant, so it is not possible to change the atmosphere, but the lung will change its intra-alveolar pressure. When that intra-alveolar pressure is more positive compared with outside, or when it is more than the atmospheric air pressure, then the air will flow out. And when the intra-alveolar pressure is lower than the atmospheric pressure, then the air will flow from the atmosphere into the inside or the intra-alveolar space. So this will be changes of pressure that are caused by the movement within the thoracic cavity. So how does the body changes the pressure in the alveoli? Well, these changes of pressure are caused by changes in the volume. Basically, the lung volume can change. It could be expanded, and also it could be compressed. When it is expanded, then the volume is increased, and when the volume is increased, the pressure will decrease, and that decrease in pressure will cause the air to flow into the lung in inspiration. While when the lung is compressed, then the volume is reduced, and when the volume is reduced, the pressure is increased. When the pressure is increased, the air will exhalate or expirate out of the respiratory system into the atmosphere. So the lung has the ability to change its volume, and by changing its volume, it changes the pressure, and that change in pressure will make inspiration or expiration possible. So the changes in the volume, as shown in this figure, let's use the pointer. So here there are basically two principal movements which are possible in the lung. One of them is the anterior posterior diameter change. So the lung, we all know now, while we are breathing, we will see that our rib cage or the chest moves upward and anteriorly. This movement is a process of expansion. It happens before the air flows into the chest. This movement is happened due to the muscles contractions, which are responsible for the expansion or the expansion of the rib cage. In this case, one of them is the external intercostalis muscle, which when it contracts, it increases the anterior posterior diameter by moving the rib cage forward and superiorly with elevation. So this movement increases the anterior posterior diameter. Another mechanism by which inspiration works is by the contraction of the diaphragm. Now, when the diaphragm contracts, it moves downward, and when it moves downward, it will increase the superior inferior diameter. So by these two muscles, the superior inferior diameter, anterior posterior diameter will increase, increasing the volume inside the lung, making the pressure low. And when the pressure is low, the air will flow from outside atmosphere into the intrathoracic space. And this, of course, will be followed by expiration. Now, in the process of expiration, usually what happens is that these muscles which have contracted, causing expansion of the intrathoracic cavity, will relax. Once they relax, then they will compress the lung, and the lung volume will be reduced. This reduction in the intrathoracic volume will cause the intrathoracic pressure to increase. That increase in the intrathoracic pressure will push the air from intrathoracic space into the atmosphere, and that is the expiration process. So this figure here shows you the basic function of the respiratory system, the inspiration and the expiration. So here you can see the movement of this. This is a bottle which is sealed, and then there is a pipe which is open, allows the air to move in and out. And this pipe represents the trachea, and it is connected to two minor divisions which are resembling the bronchi. And then there are two balloons which are attached to it. So here the bottle, the floor of this bottle is not glass, it's a rubber that could be is flexible and can be changed. So in this example, when you pull that diaphragm, it resembles the function of the human diaphragm muscle when it contracts. So it will move down, increasing the superior inferior diameter, and by this will make the volume increase, making the pressure low. So that pressure, when it is lower than outside, the air will flow into the glass jar and causing the inflation of the balloons which are attached to the two bronchi, or these pipes which resemble the bronchi. And what happens in expiration, when you leave it back, and that's what happened in the normal respiration at rest, when relaxation happens, this will compress, reducing the volume, increasing the pressure inside that jar, compressing the balloons, causing the air to flow out through expiration. So this example here explains to you how inspiration and expiration work inside the lung tissue. So, so here you can see in the figure, as we just mentioned, the movement of the mechanism of inspiration is caused by the increase of volume in the intrathoracic space, which is by two movements. One of them of the chest, the rib cage, which moves upward and forward by the contraction of the external intercostalis muscle, while the anterior posterior diameter increase by the contraction of the diaphragm, which moves down when it contracts. So these two muscles contraction will cause the increase of volume, and it will cause inhalation of air due to the decrease of pressure inside. But when these muscles relax, so the chest will retract back due to its elasticity, the diaphragm will relax and move back, upward, and this will reduce the volume in the thoracic cavity, causing the air to flow back again through expiration out from the upper respiratory tract. So these are the two movements, one in the diaphragm and one in the rib cage, altering the volume and pressure. So as we mentioned, in our now when we are sitting at rest, normal, quiet breathing, the respiratory muscles only are involved in the process of inspiration. And these muscles are the diaphragm and the external intercostalis muscle. When these muscles contract, then the volume increase and causing inspiration, while the expiration is passive process, which means it doesn't require energy, it doesn't require muscle contraction. It depends on the elastic recoil of the lung when the muscle relaxes. But this happens only at rest. Normal, quiet breathing. Under stress, when more frequent and deep breathing is needed, then both of the inspiration and expiration become active. Not only the two muscles, which we have mentioned in respiration at rest are involved. Another group of muscles are going to be included to make the inspiration more fast, more powerful. And another group of muscles are going to help in the expiration. Instead of being depending on the elastic recoil. Yes, so inspiration needs energy. Yes, inspiration needs energy. Inspiration depends on muscle contraction that requires energy, while expiration is a passive process only at rest. When we are at rest, the expiration does not require muscle contraction. But when we are in exercise, then the breathing is frequent and more deeper, needs more power. Therefore, more muscles are going to be included in that process. And I'm going to show in the next slides those muscles. So in active breathing, both inspiration and expiration is active, while at rest, only inspiration is active. So the muscles of respiration, as we mentioned before, the primary muscles are the diaphragm and the external intercostalis muscle. These two muscles are inspiratory muscles. When they contract, we exactly mentioned the mechanism, how they alter the volume of the lung, the pressure, causing inspiration. In addition to these muscles, during stress or exercise, another group will help them. These include the sternocleidomastoid, anterior serrati, scalene, and pectoralis minor. And now all these muscles will work on the rib cage, making the movement of inspiration faster by moving the rib cage through elevation and forward movement. The same action of external intercostalis muscle. Okay, while the movement of the diaphragm is not assisted in active breathing. Now in expiration, it is at rest and it is a passive process which doesn't require any muscle contraction. It depends on relaxation. But during exercise or during stress, when the respiration is fast and need to be supported, in these conditions, muscles will be used. And these muscles that are used are the abdominal recti. Now the abdominal recti will help to make the relaxation of the diaphragm faster. They will push the diaphragm up by increasing the intra-abdominal pressure. And the internal intercostalis muscle, which will work opposite action of the external intercostalis muscle. Internal intercostalis muscle will pull the chest rib, the rib cage back downward and posteriorly, compressing the lung. So, but these are not used at rest. They are used only in exercise when the high or rapid breathing is required. So this figure here shows you the same thing, the accessory muscles and the principal muscles in inspiration and expiration. External intercostalis, the diaphragm here, the principal, while the accessory sternocleidomastoid, scalene, pectoralis minor. While here in expiration, in quiet breathing, is the passive, depending on the elastic recoil, without any muscle contraction. But in active breathing, then we are recruiting the internal intercostalis, the abdominal recti, which are mentioned before. So what are the pulmonary pressures which we have already described? These pressures which will make you understand the movement of air. One of them is known as the atmospheric pressure. Atmospheric pressure is the pressure of the atmosphere, the environment surrounding us, which is roughly around 760 millimeter mercury at sea level. When we move above sea level, that atmospheric pressure will become less. Moving below sea level will cause the increase of atmospheric pressure. And that pressure the comparison between atmospheric pressure and the intra-alveolar pressure will determine whether air is going to move in or out from the lung. So the intra-alveolar pressure is the pressure inside the alveoli. When this pressure is higher than atmospheric pressure, the air will flow out. When this intra-alveolar pressure is less than atmospheric pressure, then the air will flow in from the atmospheric pressure to the alveoli. Another term, intrapleural pressure, which is the pressure inside the pleural space we mentioned about the structure of the pleura, which consists of visceral layer, parietal layer, and there is suction. And we mentioned it is always in negative pressure. It's always in suction, holding the lungs tissue within the thoracic cavity. So this is also going to participate in the process of ventilation, because some of the disturbances which are going to be mentioned at the end of the lecture, which are related to disorders of the pleural space, will affect the process of ventilation. The last one, transpulmonary pressure, is a concept related to both intra-alveolar and intrapleural. I think you just need to focus on the atmospheric, intra-alveolar, and the intrapleural pressure. Those three are important. Intra-alveolar pressure is the pressure which is of air inside the alveoli. Inside the alveoli, which are the air sacs, the air which is there, it has a pressure. That is known as the intra-alveolar, inside the alveolar pressure. This pressure, when it is higher than the atmospheric pressure, then the air will move from the lung to the outside. When this intra-alveolar pressure is low compared with the atmospheric pressure, then the air will move from out to the alveoli through the upper respiratory tract. So the main two are the first. The three are important. The first two are very important. The third one is also important. But the last one is less important to know, because transpulmonary pressure is the pressure across the rib cage, which depends on the difference between the pleural pressure and the alveolar pressure. Okay, but you can focus on the first three: atmospheric, intra-alveolar, intrapleural. You're going to see them in the next few figures. So atmospheric pressure, as we mentioned, 760 millimeter mercury at sea level, and this atmospheric air, which has this level of pressure, is a mixture of gases. Oxygen percentage is 21%, nitrogen is 78%, the majority is nitrogen, and carbon dioxide has very low level, 0.04, while we have some other inert gases which are not going to be mentioned. Okay. So as we mentioned before, the pleural space, the pressure inside it is always negative. Pleural pressure is normally in suction, and that means it's always lesser than the atmospheric pressure. Atmospheric pressure is 760, so the level of that pressure at rest, if we are not breathing, or during expiration, is -4. So that's the highest level it could reach, which is negative. During inspiration, where the thoracic cavity expands, the rib cage moves forward and upward, the diaphragm moves down. This will cause the expansion of the pleural space. So the pleura will expand with the thoracic cavity, and when it expands, it will pull the lung tissue with it to expand. So this will increase the negative pressure to more negative. So it will reach to -6 millimeter mercury below the atmospheric pressure. That is equal to 754 millimeter mercury. So 6 less than 760, that's 754. So these are the changes in pleural pressure, which is, as you can see, is always negative, from less negative to more negative during inspiration. While alveolar pressure, right now, if you open your mouth, so why the air go outside if it's still less than? The air will move outside because of the changes of pressure in the alveolar. As we mentioned, the pleura is always in negative for a purpose, to hold the lung tissue. The lung tissue, both lungs are held by suction by the pleura. But where air is going in or out, that's into the alveoli. So the alveolar pressure, that is the one which will determine whether air is going in or out. Alveolar pressure is going to follow the pleura. When the pleura expands, the lung will expand, and the alveolar pressure will become more negative. So here, if you are, if the mouth is open and you are not taking any breath in or out, okay, then the pressure inside the alveoli is equal to the atmospheric pressure, 760, and that's why there is no air movement. Nothing in, nothing out. The numbers, no, just remember the values, whether they are higher or lower, okay? You need to understand when it is less or it is more, okay? So here the alveolar pressure when we are not breathing is exactly the same as atmospheric pressure, 0. While in normal inspiration, so in order for inspiration to happen, of course, the intra-alveolar pressure will become more negative due to the expansion of the thoracic cavity by the muscle contraction. Of course, the pleura will expand with it. It's not going to expand, it's going to be pulled by the rib cage, and this will make it more negative. And then this will make the lung tissue expand, pulled with the pleura, making the pressure more negative, which is -1 millimeter mercury compared with the atmospheric pressure. This slight negative pressure is enough to take ½ liter of air from the atmosphere into the lung through inspiration, which will last about during 2 seconds. Okay, this is normal quiet breathing. Then, when the muscles relax, the rib cage compress the pleura will compress on the lung tissue, the negativity will get less, and the alveolar space will compress, causing positive pressure. So that positive pressure is 1 millimeter mercury higher than the atmospheric pressure, which is enough to push back that ½ liter back through expiration to the atmosphere, which takes slightly longer time, 2 to 3 seconds for full expiration at rest. Okay? So here this figure shows you the whole thing together, shows you the change of the pressure during inspiration and expiration. You can see here the alveolar pressure in relative to the atmospheric pressure. So it is -1 during inspiration, +1 during expiration. And here in inspiration, ½ liter in, and during expiration, ½ liter out. So this is again the same thing, so we're not going to explain it. So at this point, we understand which muscles are involved in the process of inspiration, what happens in quiet breathing when we are at rest, and what happens when we are in exertion or we are doing exercise. So what are the other groups of muscles which are going to be recruited, and how does this muscle contraction helps to change the pressure, and what are the values of pressure that are going to change, causing the air to move into the lung or out from the lung. So until this stage, then the next is going to be control of respiration. What is the center that controls respiration? So normally breathing controlled by contraction and relaxation of the diaphragm and external intercostalis muscle, as we mentioned. Although the respiratory muscles can be controlled voluntary, normal breathing is rhythmic involuntary. So these muscles, the external intercostalis muscles and the diaphragm muscles, are skeletal muscles. These muscles are both voluntary and involuntary. And in my opinion, they are more involuntary than they are voluntary, because you can control them, but to large extents, they are moving involuntary depending on the impulses sent from the respiratory center. So they can be controlled voluntarily, but largely they are working involuntary, causing the process of inhalation followed by exhalation. Now these contractions, which are involuntary, happen due to the impulses coming from the respiratory center in the nervous system. In the central nervous system, in the medulla oblongata, we have the respiratory center, which is shown here in the figure. So here, this is the brainstem region, which is the in the beginning of the spinal cord. So here, this is the cerebellum, and here, this is the brainstem, which consists of the lowermost part known as medulla oblongata, followed by the pons, and lastly the midbrain. This is the brainstem region. So we have three groups of neurons that are working to control the respiration process. Two of them are located in the rhythmicity area in the medulla, and one of them is in the pons, known as the pneumotaxic area. All of these are very important to control the process of respiration. So, the most important group, and I'm sure if you are going to refer back to your textbook, you will find it different. So I'd like you to focus here to follow the slide. The primary group of respiration that works at rest is the dorsal respiratory group. Dorsal respiratory group is the part of the respiratory center that controls the contraction of the diaphragm and the external intercostalis muscle. And this group, therefore, we depend on it in quiet breathing. While the ventral group is the group used under stress while we are in physical activity, and this ventral group will be responsible for the accessory muscles that were being mentioned before. We have mentioned about the sternocleidomastoid, the scalene, pectoralis minor, the abdominal recti, the internal intercostalis, those accessory muscles, they will be contracting in response to the ventral respiratory group. So in normal quiet breathing, we are under the control of the dorsal respiratory group. So the dorsal respiratory group in the medulla oblongata, in the rhythmicity area, is responsible for the basic rhythm of breathing. Neurons. So breathing ventral controls. The ventral group controls the accessory muscles in inspiration and expiration, while the dorsal respiratory group controls inspiration in the quiet breathing. As we all mentioned, the expiration in quiet breathing is passive process. It doesn't require muscle contraction. The muscles will relax and then expiration will happen. So the dorsal respiratory group is the primary controller, or the basic, responsible for the basic rhythm of breathing. And in this group, the neurons, the dorsal group, yes, they are responsible for inspiration, the diaphragm and the external intercostalis muscle. So the neurons of this group will send the nerve impulses to the diaphragm and the external intercostalis muscle, while the ventral group remains totally inactive during normal quiet breathing. We don't use them in normal quiet breathing. When under stress, when frequent forceful breathing is required, then we start using the ventral group, which will start sending signals to inspiratory movements in addition to additional expiratory movements. Yes. No, both of them are the same. Dorsal and ventral, both of them, they are voluntary and involuntary, but they are largely involuntary. They work only when they are required in exercise or in stress. Okay, so both of them, they are the same. Both of them are responsible for control of respiration. The dorsal group controls only inspiration, while the ventral group controls inspiration and expiration only during exercise when forceful breathing is required. So the primary control of breathing at rest is the dorsal group of neurons in the medulla oblongata. Now we understood those two. How about the third group, which is located in the pons, that is known as the pneumotaxic neurons? this center of pneumothoracic neurons, their function is to control the respiratory rates. How do they control the respiratory rates? They send inhibitory impulses to the inspiratory area. So in the inspiratory area, the dorsal group in quiet breathing and the ventral group together with the dorsal group in active breathing, they are responsible for the inspiration. So the pneumotoxic area will terminate those groups to stop the process of inhalation. So these inhibitory impulses will shorten the duration of inhalation. So the inhalation is by the dorsal group, and they will be stopped by the pneumotoxic neuron center. So when the pneumotoxic neuron center is more active, this means inhalation is going to become shorter, which in other words, the breathing is going to become more rapid because each cycle of breathing is shorter. So instead of having a breathing of 20 cycles per minute, when the pneumotoxic area is active, it might become more frequent, up to 25 or 30 cycles, and especially in exercise where the respiratory rate is higher than the normal range. So the pneumotoxic area will set the respiratory rate by sending inhibitory impulses to the inspiratory area situated in the medulla oblongata, determining the respiratory rates. So these are the three groups: the pontine group, the ventral, and the dorsal group that are working together in order to facilitate the process of inhalation and exhalation. So inhalation through the intercostal nerves and the phrenic nerve to the diaphragm and external intercostalis muscle in the dorsal group, while the ventral group here through other nerve fibers to the internal intercostalis and other groups of muscles. Okay. Neural control of breathing. So the respiratory center, we just explained it. It is the neural control of breathing. It controls the muscles by the nerves. These nerves are the phrenic nerve that supplies the diaphragm, the intercostal nerves that supplies the external intercostalis muscle, which will control the expansion of the rib cage and the movement of the diaphragm that will increase the volume inside the thoracic cavity, making the pressure negative during inspiration. And when the signals stop from the phrenic nerve and the intercostal nerves by the inhibition of the pneumotoxic center, then the muscles will relax, causing a process of expiration. The expiration, which is at rest, relaxation, not without muscle contraction. So the normal respiratory rate for an adult is between 12 to 20 cycles per minute. There is one characteristic in the signal that is generated in the respiratory center. That signal has a characteristic which makes it have the name of a ramp signal. In this regard, because the diaphragm and external intercostalis are skeletal muscle, and like skeletal muscles, usually their contractions are supposed to be instantaneous, fast. But here, if this same contraction happen in the diaphragm and external intercostalis muscle, this will cause the rapid expansion of the thoracic cavity, making gasps of air. So we don't normally have this process of ventilation. Normally, the signal is a ramp signal, which starts as a slow and gradually increase, making muscle contraction, gradually expanding the thoracic cavity and making the flow of air gradually into the thoracic cavity. So due to this ramp signal, which starts as a low signal, gradually increasing, the ramp signal makes, I think I better show you in the figure. So you can see here the ramp signal that goes into the respiratory muscles is gradual and increasing, making the expansion of the thoracic cavity gradual. So these muscles will not contract fast. They will contract slowly by the ramp signal generated from the respiratory group of neurons in the respiratory center. So this will make a gradual expansion of the chest, so a smooth inflation of the lungs, and there will be no gasping by this ramp signal. I hope it's clear now. So normally each cycle will take 5 seconds. That's why in 60 seconds we will have around 12. Yeah. So 3 seconds for expiration and 2 seconds in inspiration. The expiration takes longer time. So when we gasp, that means we are the expansion is fast. So that's the gasping. The rapid inhalation of air, which normally does not happen. Normally, the expansion is gradual by the generation of the ramp signal, which will make the breathing gradually expanding, and the air will smoothly fill up the thoracic cavity and the lungs. Now, our respiratory rate, is it fixed, or it can change? Of course, we know that sometimes we can know that when we are sleeping, for example, the respiratory rate is slow. When we wake up, what is the difference between the two photos? You mean these two? This one is representing the ramp signal. That is showing the movement of the air in and out. Yeah. So the volume here of the half liter of inspiration, expiration. So here, this is the timing of them together. So here you can see the signal within the phrenic nerve, okay, over 2 seconds in inspiration, causing the inflow of air. Then when this stops by the pneumotaxic area, then the air will move out and there will be no more signals. So this is the duration of 3 seconds of expiration. Then the cycle will start back again. So these are the 5 seconds. So yes, they are not the same. One of them, the upper one, is about the movement of air, the volume of air in and out, the volume of air, while the lower one is about the neuron activity, the nerve activity of the phrenic nerve, which is, this is the ramp signal. The lower one is the ramp signal. The upper one shows you how the ramp signal gradually makes the air go in. You don't have a sudden spike of air going into the lung. Instead, the air moves gradually until it reaches the half liter. And it is, when it is expired, it gradually moves out. Okay? So, what are the factors controlling the respiratory center? Now we know that in certain conditions, our breathing could be slow when we are sleeping. Sometimes when we are running, exercising, then our breathing is fast. Now this definitely means that the impulses which are coming from the respiratory center are not constant. They change. And this change happens in response by other multiple factors. One of these factors is the chemosensitive areas. Now, chemosensitive areas are located inside the respiratory center. Now, the respiratory center, which we have just described in the medulla oblongata, it's responsive to the chemistry in the blood. Most importantly, CO2 and hydrogen, not oxygen. So, if the question comes, the primary or the main factor that controls our respiration, the answer is CO2, then hydrogen, and not oxygen. Oxygen is not the primary controller. So let's say, for example, if you have hypoxia and at the same time you have low level of CO2, then your body will respond to the low CO2 and you will not feel you need to take a breath. Although oxygen is the more important. but our respiratory system responds mainly to the changes of carbon dioxide, not oxygen, CO2 and hydrogen atoms. Of course, carbon dioxide inside our body will change into acid, and it will make hydrogen ions by making carbonic acid. So CO2 and hydrogen atoms are the main controller of respiration by the chemosensitive areas, the central chemosensitive area within the respiratory center. So central chemoreceptors will respond to the changes of CO2. So when CO2 is high, then the respiratory centers are going to be stimulated in a way to increase the respiratory rate. And once the respiratory rate is increased, this will wash out the CO2. And when the CO2 is low, this will make the respiratory center less active, so the breathing will become slow. So while we are sleeping, for example, the metabolic rate is very low and the level of CO2 is low, therefore our sleeping is, the breathing while we are sleeping is slow. But when we are exercising, running, in the beginning, the breathing will not change. But once the CO2 levels increase in our circulation, then this will stimulate the respiratory center, making the rapid respiration or tachypnea. That's what happened when we are exercising. That's the reason why we breathe fast when we are exercising, due to the generation of CO2 by the high metabolic rate in the skeletal muscles. So the low blood oxygen have little effect on the chemosensitive areas. But there are other receptors that could be influenced by the level of oxygen, and these are the peripheral chemoreceptors situated in the carotid artery and the aortic arch. Now these are responded to low oxygen and can stimulate the respiratory center, but their influence is less compared with the central chemosensitive areas situated inside the respiratory center. Therefore, this should not be used in order to prolong the diving time while swimming. Some divers, for example, they can hyperventilate to wash out the CO2, and once they wash out the CO2, then this will inhibit the respiratory center, so they will not feel they have to take a breath, and they can dive for a longer time below the water. And this is very dangerous because while you are hyperventilating, you are washing out the CO2 and washing out acid, which can cause alkalosis. And at the same time, while diving, the drop in the level of oxygen will not cause the person to breathe, because the overwhelming low CO2 will influence the respiratory center, and the person will not feel they have to take a breath, although the oxygen level is low, which is very dangerous, both alkalosis and hypoxia below the water, which can cause loss of consciousness and drowning. So other factors which control breathing, we have just described the central chemoreceptors in the respiratory center. I did not understand. While diving, some people can use a mechanism to prolong the time they can dive under the water before they go up to the surface and take another breath. So by hyperventilating, by breathing fast, washing the CO2 out from the circulation, causing the level to go down. So by that mechanism, the CO2, in order to rise up to the significant level, will take longer time. For that reason, the diving can be longer time below the water. So it is very dangerous, because that maneuver is associated with hypoxia and alkalosis. So both conditions, hypoxia and alkalosis, will cause loss of consciousness to the diver, which is diving below the water. So the attempt of hyperventilation in order to wash out the CO2 and increase the diving time is dangerous. No, it's not bigger lungs, but fast breathing that will wash out the CO2 and causing alkalosis and hypoxia. the alkalosis and hypoxia is the two dangerous factor that might cause loss of consciousness while diving into the water. So what are the signals or the factors that control breathing? We just mentioned the central chemosensitive area, which is number one. The peripheral, we also mentioned about them in the carotid and the aortic artery. Then we have some other receptors situated in the walls of the lungs. When the lung is overinflated, then they will send inhibitory signals to stop inhalation, stretch receptors that will prevent overinflation of the lung. Another factor is presence of irritant receptors. These mainly can cause cough. Coughing is by the irritant receptors, which are mainly present in the trachea, the carina, the larynx. So these in the upper respiratory tract and the upper lower respiratory tract can cause cough. Pain and proprioceptors in the muscles and joints can also increase respiration. Emotions, you have, I'm sure you have seen some movies whereby some people, when they are in extreme fear, then this extreme fear is usually associated with rapid breathing when they are afraid. So that's why how the you see that the limbic system or the emotions can also change the respiratory center signals, making breathing faster. And of course, the lastly, breathing could be changed consciously with consciousness. So with that, we have completed the muscles of respiration, the mechanisms of respirations, the changes of pressure, the respiratory center, the nerves which are responsible, the characteristic of that impulse. We have described also the regulators of the respiratory center, the chemosensitive areas, the other factors that could control the process of breathing. Now we are reaching a new term known as the compliance of the lungs. So this is important. Compliance of the lung is, I will explain it first in simple words, and then we will read. Compliance of the lung is a term that describes how easy the lung can expand upon inspiration. So a lung compliance is the magnitude of change of the lung volume produced by the change in pulmonary pressure. When the compliance of the lung is in good condition, that is a lung with high compliance, this means that you need less energy in order to expand the thoracic cavity. Okay, so the lung tissue itself is flexible and it is elastic. It could be easily expanded with less pressure change. Okay, this means a lung with good compliance. So compliance is considered the opposite of stiffness. Why? Because when the lung is stiff due to a disease, it becomes more difficult to expand. When it is more difficult to expand, it will need more and greater than average change in the intrapleural pressure or effort in order to change the volume of the lungs. So that's why lungs with low compliance are more difficult for breathing. We are going to explain some of the most important disorders nowadays which are related to it. This has come with practicing? No, not with practicing. They are related mainly to two factors, and both factors cannot be changed. One of the factors is the elasticity of the lung, so how much elastic tissue is there and how much collagen tissue and fibrosis is there. If there is more elastic tissue, less collagen tissue, then the lung tissue is elastic and easily to expand. Okay, this cannot be changed. And another one is the surfactant, which affects the surface tension. We explained it before. The surfactant, which is released by the alveolar cell type 2. Not age, but due to diseases. Diseases with fibrosis. Now, we will explain it later. We will explain some of the disorders that will interfere with the compliance of the lung, but mostly diseases. Diseases that will make the thoracic cavity more difficult to expand on breathing. Asthmatic people, they have nothing wrong with their compliance. The problem in asthmatic people is related to the bronchioles. I will explain it later. We will talk about this in more details. Just now, understand that the compliance of the lung is how easy that lung can expand in the process of inspiration. A lung with high compliance is a good lung, easy to expand, while a low lung compliance is a stiff lung that requires more energy in order to expand. It needs more pressure and more effort in order to make the volume changes. And as I mentioned, the two factors that will determine the compliance are the elasticity of the lung and the surface tension, which is controlled by the surfactant. We have already explained the role of surfactant and where it is secreted and how it works. So we are not going to explain this again. Those are some of the disorders that are related to deficiency of surfactant, which we explained one of them in the babies, infant respiratory distress syndrome, which happen due to increased surface tension by the deficiency of surfactant. And they will develop collapse of the alveoli because of the increased surface tension as shown here in the figure. So this is an RDS in a baby, which is immature. And the left side shows a normal alveoli, which can be fully inflated. So lung collapse happens when there is a deficiency of surfactant, causing the decrease in the compliance of the lung. The lung becomes more difficult to expand. And that's why those babies are kept on ventilators in order to inflate the lungs in inspiration. This is another picture of a baby with respiratory distress syndrome. So next we will reach to the lung volumes and capacities. I'm sure you have described it in the practical sessions, the spirometry, how you measure the lung function tests. So what is important here? Have you explained this figure in the practical session? You understand it? Did you understood it well? So I don't need to explain it again to you. So this is the tidal inspiration, tidal expiration, which is 500 ml of air. And this is what we are doing at rest. Now, if we inspire the 500 ml of tidal volume, fibrosis increase compliance? No, the opposite. Fibrosis will reduce the compliance. Fibrosis is bad. So it will produce a poor compliant lung, more difficult. Fibrosis is going to make it more difficult for the lung to expand. When it is more difficult, that means the lung is stiff, the lung is having poor compliance. Because the compliance is how easy the lung can expand. And this is the opposite of what happens when there is fibrosis. So, inspiration here in the tidal volume, 500 in, 500 out, 500 in, 500 out, tidal volume. So this is the tidal respiration, both inspiration and expiration. Now, is that all the amount that we can inspire? No, we can take a maximum inspiration, and that maximum inspiration after tidal inspiration, starting from here, that's amount of air, 3000 ml or 3 liters, is inspiratory reserve volume. Reserve means we don't use it normally. Yes, elasticity is good. So fibrosis is mainly by the deposition of collagen fibers, which is bad for the expansion of the lung. So you can see here the inspiratory reserve volume, which is the 3 liters of air that could be taken in inspiration. Now, That amount, if we want to expire it, we can take it out, the inspiratory reserve volume, and expire in addition to it the tidal volume. But is that all what we can move out from the lungs? No, we can move also an additional volume for full expiration. This is known as the expiratory reserve volume. So this is only the expiratory reserve volume. So there is a certain amount of air which remains inside the lung. That amount is the residual volume. That amount could not be measured in spirometry. That amount will never move out from the lung. Otherwise, all the alveoli will collapse. So there is a certain amount of air that is left in the alveoli, and that amount is 1,200 ml. This if we have done maximum expiration. But right now, at rest, we have the functional residual capacity, which is 2,300 ml. That 2 liter and 300 ml remains inside the lung when we are breathing tidal volumes. In inspiration, expiration, okay? So we have 2,300 ml. I want you to understand this figure very well because number of questions in the MCQs are usually from this figure. So you need to differentiate between the amount of air inside the lung and the amount of air which is inspired when we are explaining the scenario of the question. Sometimes I will tell you the amount of air which is inspired at rest, that amount is tidal volume. If I'm telling you the amount of air inside the chest after tidal inspiration is, then that includes this tidal volume plus the expiratory reserve volume plus the residual volume, because that is the amount of air which is right now inside the lung with tidal inspiration. If I tell you what is the amount of air inside the lung after maximum inspiration, then you need to know everything. Number, no, but you need to know it is including all these three. 1, 2, 3, 4. So this is the amount which is the total lung capacity. So the amount of air inside the lungs after maximum inspiration is the total lung capacity. But the amount of air inspired after tidal inspiration is only the inspiratory reserve volume. The amount of air that is maximally exhaled after maximum inspiration, what is that? That is the vital capacity, because you will exhale this part, and this part, and all of them together represents the vital capacity. So you need to understand each one of these in order to understand which one is the right option for the right question in the multiple choice. So here the explanation I'm not going through. I have already explained it to you, and you have already taken it in the practical session. You don't need to remember the numbers, only the tidal volume, you need to remember it, which is 500 ml. So now we reached to the cuff reflex. Cuff reflex is one of the protective mechanisms regulated by the medulla oblongata. And as we mentioned, there are irritant receptors in the larynx, trachea, and carina that are stimulated by irritants. So these could be infectious materials, could be particles. Sometimes if there is some dust, then it will make you cough. So these irritant substances will stimulate the receptors, and they will cause the process of cough, which with the steps which are shown here. So the diaphragm and the external intercostalis muscle will contract, causing inhalation of air, not the tidal volume only, but a larger amount, but not the maximum, around 2.5 liters of air. And then once that pressure is inside, then immediately the glottis and the vocal cords will close, trapping that air inside. So at the same time, the abdominal muscles will contract, the expiratory muscles will contract, and then suddenly all of this will increase the pressure very high inside the chest, and then the vocal cords will open. When they are opened like that, this will make the air move with a very fast speed, around 100 miles per hour, and this high speed will push out all the irritants from the respiratory system to the outside. So during that stage also, the non-cartilaginous portions of the trachea and the bronchi will contract, making slits that will expose the particles, the mucus that will be pushed out, and that's why sputum is released in the cuff. Yeah. Sorry, can you repeat the question? Okay, how long it is? The cuff reflex? So seconds. I'm sure you are aware about cuff. We all cuff, so you know that's how cuff happens. So you know how long it takes. So other non-respiratory movements are crying, laughing. This will usually accompany the emotional conditions, including the limbic system. So inspiration followed by release of air in number of short expiration. Laughing is similar. Hiccups, for example, is sudden inspirations due to contraction of the diaphragm by the full stomach that usually produce sounds when the air moves through the vocal cords. Yawning is deep inspiration with jaws wide open while people or the person feels sleepy, especially with the drop of the blood oxygen levels. The dead space, we have explained it. We mentioned that when we breathe, we take around half liter, 500 ml. From this volume, 150 ml is going to be in the conducting zone, not playing any role in the gas exchange, while the other 350 ml is going to reach the alveoli and participate in gas exchange. Anyone can tell me now in normal quiet breathing how much air is there in the alveoli? How much volume before the 350 ml reach there? No, the 150 is in the upper respiratory tract. Muzdalifa is very close. She mentioned about the residual volume, but in normal breathing, residual volume plus the inspiratory reserve volume. Sorry, the expiratory reserve volume. So together is 2,300. That is the functional residual capacity. 2,300 ml. This is the amount which is inside the lung before we take any breath, when we are at rest. So the 350 ml will mix with that 2,300. So when we are breathing, this doesn't mean that you are changing all the air inside the lung with every breath. You are changing a small volume of the bigger amount of air which is remaining there inside the lung, which is the functional residual capacity. 2,300 ml mixes only with 350. The percentage is 1/7, seventh of the volume. Okay, so what are the functions of the dead space? Mainly they are the same functions of the conducting zone, which we have already explained them. Cough reflex, sneezing reflex, ciliary movement, filtration, conditioning, immune function, olfaction, smelling, which is in the nose, vocalization, which is in the larynx. So these are all what happens in the conducting zone. So this is what we have mentioned, the functional residual capacity of 2,300 mils, and this will mix with 350 mils, while the other 150 will remain in the upper respiratory tract in the dead space. So this will make it, we will need frequent breathing, several cycles in order to change the air inside the lungs. All right, so now we reach into the next topic, which is the respiratory membrane. We have already talked about it. The two layers, which are basement membrane, endothelial cells, basement membrane, alveolar squamous epithelial cells. Which is the very thin layer in the respiratory membrane, which is responsible for the gas exchange. Now, in this membrane, as you can see, there are some alveoli which do not have blood vessels over them. not all of the alveoli have blood supply. Now, these alveoli which are lacking the pulmonary capillaries are considered dead space because they are not playing a role in gas exchange. There are also some capillaries which are running without passing through the alveoli inside the lung, and this will cause pulmonary shunt, left to right shunt, which are going to cause a drop in the level of oxygen because it's not being participating in the gas exchange. Anyway, we will go into the partial pressures of oxygen and carbon dioxide. I'm sure this is too much for you, but it is important to understand how the pressure changes happen inside the lungs. So here we mentioned before, oxygen is 21% of the atmosphere. The atmospheric pressure is 760. So if we do a simple calculation, then we know that the partial pressure of oxygen in the atmosphere is 160. Now that air, when it is going into the lung, is going to mix with the functional residual capacity. So the percentage or the level is going to drop from 160 to 104. Then this level of 104 is going to be playing a role in gas exchange with the pulmonary capillaries. While carbon dioxide in the atmosphere is 0.04, if we calculate it, it's going to be 0.3 millimeter. But this volume, because it's going to mix with the large volume of air inside the lung, the functional residual capacity, then do we have to do these calculations? No, but you need to know the level. The one which is highlighted in green, you have to know it. The PO2 of air inside the lung. The PCO2 inside the lung is 40. So 104 and 40. So the oxygen in the alveolar air is 104. The PO2 of blood, P is partial pressure. Partial pressure, okay? So the partial pressure of oxygen inside the lungs is 104. And the same pressure for oxygen inside the veins, okay, is around 40 millimeter. Yes, the lecture will be uploaded. Yes. Okay, so the PO2, which is around in the arterial blood is around 90, and it will drop from 104. I will explain it. So in the alveolar air is 104, while the blood which is coming to the lung deoxygenated, the level of PO2 is 40. So first the level will reach 104, but then it will drop to 97 due to the pulmonary physiological shunt, those which are not included in the gas exchange. And then the level will drop further to 95 due to the physiological shunt in the heart, due to the presence of the veins within the walls of the heart. So that's for oxygen. So oxygen is between, in the arterial side is 90 millimeter mercury, while in the venous side is around 40. In the lungs is 104. While CO2 is in the venous blood, which is deoxygenated, is 46. In the alveolar air is 40, and in the arterial side is 40. It reaches the same level, and it is not affected by the physiological shunts. And CO2 is the main controller of respiration. As we mentioned before, CO2 has a very good diffusion capacity compared with oxygen. So here it shows you the changes of pressure that happen in oxygen and carbon dioxide throughout the cardiac cycle. So here it is 104 in the alveoli, or 105, okay? And then the oxygen will reach to that limit, but due to the physiological shunts, it will reach to 95. Then this level will reach to the left side, and it will be pushed out to the circulation. And after gas exchange, it will drop into 40. It will go back to the lung for gas exchange. And the PCO2 comes as 45, After gas exchange, it reach 40, and after the tissue exchange, it will go back to 45. That will go back to the lungs for gas exchange. So you need to know the three numbers for each gas. For CO2, 40 and 45 has two readings, while for oxygens, 104, 95, and 40 millimeter of mercury. So one important thing to mention in oxygen transport is that majority of oxygen is transported in the blood bound with hemoglobin, okay, which is inside the red blood cells. So this is the majority of oxygen. While how much is the amount that is dissolved in the plasma? It is only 1.5%. So how is the oxygen carried in the blood? It is carried bound with hemoglobin. That's the main portion, 98.5. While in CO2, the percentage are different. Mainly CO2 is carried out as bicarbonate. We will explain it later. So oxygen transport in the blood. How many red blood cells do we have? Roughly 20 to 30 trillion red blood cells. Each blood cell has nearly 270 million hemoglobin molecules. You don't have to remember these numbers, but just describing to you how many hemoglobin molecules do we have. And these hemoglobin molecules, the main function of them is to carry oxygen. And in the structure of the hemoglobin, they have these 2 alpha, 2 beta chains in their quaternary structure, which has also iron atom, which binds reversibly with oxygen. And hemoglobin is the perfect carrier for oxygen. Without hemoglobin, the human body will not be able to survive. First of all, what is that? Why is the reason for that? Why is it the perfect carrier? This is because hemoglobin increases the total blood oxygen capacity. So our blood can carry more oxygen, 70 times more compared with dissolved status. We can carry much more oxygen with hemoglobin than as a dissolved oxygen in the blood. That's number one. Number two, the hemoglobin affinity to oxygen is not fixed. Hemoglobin combination with oxygen can change according to the environment it is inside. Let's say when hemoglobin is in the lung, it will tend to bind with more oxygen molecules. So its affinity will increase to the oxygen molecules. And when when the hemoglobin is in the tissue level, when the PO2 is low, then in these conditions, hemoglobin will tend to release more oxygen. Its affinity will decrease. It doesn't want. So it will take oxygen when it is very high, and it will give oxygen when it is very low. So this is the characteristic of hemoglobin, which is perfect for its function. In addition, the affinity of hemoglobin can also change by other factors such as pH, PCO2, temperature. This is important, for example, when we are exercising. So when we are exercising, running, and the muscles having high metabolic rate, the muscles are producing large amounts of CO2, and this CO2 will decrease the pH. At the same time, the temperature of the body will increase. So all these factors will cause the hemoglobin to have less affinity to oxygen. And the hemoglobin, therefore, will release more oxygen to the muscle tissue, making the contraction supplied with the enough loads of oxygen that is required. So that's why hemoglobin is a perfect carrier. Now, how does that happen? This is mainly due to the protein structure, which I have just explained to you. This quaternary structure of hemoglobin is able to change when it attaches with oxygen. It changed from tense configuration to relaxed configuration. I'm not going into details, but this is how just you can know about it. How does it change? That change is in the structure of the hemoglobin from tense to relaxed configuration, depending on the availability of oxygen. In general, the relationship between oxygen and hemoglobin could be presented on a graph. And this graph is called the oxygen hemoglobin dissociation curve. Do we have to memorize this slide? Which slide? Previous one. Sorry, let's see. Understand these two terms, relax and tense configuration. That's it. You don't have to know it all. So here, the relationship between hemoglobin and oxygen is plotted on a figure. This figure is known as the oxygen hemoglobin dissociation curve. It is, you don't have to fully understand it, but just, this is the figure here. You don't have to draw it, by the way, but you need to understand what does it represent. So here, these are the saturation on the vertical axis, and here is the partial pressures of oxygen. So this lower partial pressure of oxygen is in the venous side, in tissues, while higher partial pressures are in the lung. And this relationship is a sigmoid shape. And the reason for this sigmoid shape is by the T and R configuration of the hemoglobin. You just need to understand that this curve can shift to the right or to the left depending on the situation inside our body. And for example, when it shifts to the right, okay, so I need you to understand shifting of that curve to the right means what? When that curve is shifted to the right, it means the hemoglobin will release more oxygen. And the factors that can cause this shifting to the right is the acidosis, increase in CO2, increase in temperature, as we mentioned in the muscle, increase in BPG. So these factors will shift the curve to the right, and when the curve is shifted to the right, this means the hemoglobin affinity is less to oxygen, which means the hemoglobin will release more oxygen. While when the curve is shifted to the left, this means hemoglobin have more affinity. Therefore, in the tissue level, the curve is shifted more to the right, while in the lung tissue, the curve is shifted more to the left. So shifting to the right, what does it mean? So it means something good or bad. No, both of these happen in our body. When it is shifted to the right, the hemoglobin will release more oxygen, which is required especially for muscle contraction. In the lung tissue, it's going to be shifted to the left so that the hemoglobin will have more affinity to the oxygen, and it will combine with more. So both conditions happen in our body. Both conditions are necessary. Another thing is that I will not talk further. Just keep it to this level. Okay? So the relationship between hemoglobin and oxygen is plotted in a figure known as the hemoglobin oxygen dissociation curve. When this curve is shifted to the right, it means hemoglobin will release more oxygen. The affinity will be less. While when it is shifted to the left, it means the affinity will be more. Hemoglobin will attach more with oxygen molecules. And what are the factors that shifted to the right, and what are the factors that shifted to the left, which is the opposite? Is only increase, and the left is decreased. So this is also important in the multiple choice questions. Okay? We are approaching the end now. So lastly, hypoxia. So here, what are the causes of hypoxia? So hypoxia means low blood oxygen. What are the factors that can cause hypoxia? So it is very easy to understand, maybe difficult to remember. You can see 12 points. So basically the first thing that can cause hypoxia if we are living in an environment with low oxygen. So deficiency of oxygen in the atmosphere, for example, in high altitude, or a room which has no oxygen. So nothing wrong with the human body, but the environment has no oxygen or low oxygen tension. So this can cause hypoxia. Second factor, if the atmosphere around you is normal, but then the respiratory center is depressed, it is not sending signals for respiration, then this is also going to cause hypoxia. If the air is normal around us, the respiratory center is normal, it was working, but the nerves from the respiratory center have problems due to different neurological diseases like motor neuron disease, then this is also going to cause hypoxia. These three happen outside the lung: the respiratory center, the atmosphere, and the nerves. So if the air is normal, respiratory center is normal, neurological nerves are working normal, but then we have problems in the muscles of respiration. Neuromuscular and muscular disorders, then this will cause hypoventilation and hypoxia. If the deficiency of oxygen is the oxygen of the atmosphere is normal, respiratory center is normal, the nerves are normal, the neuromuscular junction and muscles are normal, but there is decrease in lung compliance, such as in fibrosis, then the expansion of the chest is going to be more difficult, and it might cause hypoxia because it is difficult to expand the chest. The muscles are okay, but the lung is rigid. It's difficult to expand. If the air around us is normal, the respiratory center is normal, nerves are normal, neuromuscular junctions are normal, the muscles are normal, the lung compliance is good, but we have narrowing in the airways, like what happens in asthma. So in asthma, the airways, the terminal bronchioles are in spasm due to inflammation. So this will cause less air to be reaching into the alveoli. This can also cause hypoxia. Now, if air is in the environment is normal, respiratory center is normal, the nerves, neuromuscular and muscles are normal, the lung compliance is normal, the bronchioles are open, and the air is flowing into the lung, but we are having problem in the alveoli, which is thickening of the respiratory membrane due to inflammation. Then this will alter and change, make it more difficult for oxygen exchange. Then hypoxia will happen. If the alveoli are damaged, like what happens in emphysema, then this will also cause hypoxia. If there is a problem in the pulmonary circulations within the alveoli in the respiratory membrane, so then this is also going to cause hypoxia. So number 7 is mainly about the respiratory membrane. Now, if the environment is normal, respiratory center is normal, nerves, neuromuscular junction, muscles, lung compliance, bronchioles are open, alveoli are normal, and air is flowing into the alveoli, but then we have poor circulation. The blood is not flowing efficiently to the lungs, such as in heart failure. Then there will be a problem of gas exchange, and hypoxia will happen. If everything is normal, including the heart, but there is right to left shunt, this will mix deoxygenated blood with oxygenated blood, and it will also cause hypoxia. If everything is normal, the cardiovascular system is normal, but then we have a problem with the hemoglobin. The affinity of hemoglobin is changed because of, example, carbon monoxide poisoning. So the hemoglobin will not be able to attach with oxygen. Then we will also have hypoxia. If the person has anemia, which is low hemoglobin level, again, they will suffer from hypoxia, although everything else is normal because the carrier is missing, hemoglobin. And lastly, if all these are normals, but the blood vessel is closed, like what happened in myocardial infarction, this will also cause hypoxia to the tissue. Okay, so these are the different factors which are associated with hypoxia. Now we reach to this topic. We are all aware about the pandemic of the coronavirus infection around us, which is the reason why we are proceeding through distant learning sessions. So it is useful to know some information about this virus. This infection by this novel virus, it causes infection to both upper and lower respiratory tracts. But what are the causes of death due to this virus? This virus, it causes the activation of the human immune system through what is known as the cytokine storm. So this cytokine storm will make intense inflammation within the lung tissue, And that intense inflammation within the lung tissue will cause thickening of the respiratory membrane. So you can see here on the right side, the respiratory membrane is very thin, which is 0.5 micrometer. And in those patients who are suffering to death, the respiratory membrane is very thick, so it is very difficult for oxygen to pass through the respiratory membrane, and that's why they are suffering from hypoxia, and due to that reason they are kept on ventilators. Another thing also, due to the fibrosis, they have poor lung compliance. So this respiratory membrane is thick and their compliance is poor, and both of these causes are causing the hypoxia in those patients, and that's why they are kept on mechanical ventilators. We all hear about the mechanical ventilators, which are helping those patients to survive, because it is impossible for them to supply the oxygen in their breathing due to the thickening of the respiratory membrane and due to the loss of lung compliance by the fibrosis, which happens due to the infection. So we hope, inshallah, this problem will be over soon and everything will be back to normal. And we wish you best of health. Please be reminded in the precautions which are well known to you to avoid any social gathering and to intensify the hygiene of your hands in order to avoid any contamination with this virus. As we can see, this virus is very dangerous and it could lead into these disastrous consequences, especially in people who have a weak immune system, those who are elderly, heart failure, obesity. Okay, so in those people, they are going easily develop the cytokine storm and they can reach into respiratory failure and death. Okay, so that's oxygen. We reach now into the CO2 transports. CO2 transport, which is different from oxygen. Majority of CO2 is transported in the blood as bicarbonate or carbonic acid. Both of these are the same. And this is the majority of CO2. 23% lesser extent is carbon hemoglobin. So CO2 also can bind with hemoglobin. 7% dissolve. So in the multiple choice question, usually I would ask question in this regard. The majority of oxygen is transferred in the blood in the form of A, B, C, D. So you know that the majority of oxygen transferred bound with hemoglobin. CO2, the majority of it is as bicarbonate or as carbonic acid, which is the same. So this conversion of CO2 into carbonic acid is mainly by the carbonic anhydrase enzyme, which makes the reaction very fast, up to 5,000-fold more. And this enzyme makes it very, it is convenient to transfer carbon dioxide within the circulatory system. Some drugs can interfere with the function of this enzyme, which can interfere with the transport of CO2 in the blood, causing the CO2 retention and problems in the respiratory system. What are the factors that could affect CO2 concentration in the blood? Basically, the ventilation. So hyperventilation. How can we lower CO2 in our blood? Hyperventilation is the cause. Hyperventilation can lower CO2 by washing out the CO2 from the respiratory system. So increasing respiratory rates or deeper respiration, this will wash out the CO2 from the circulation. What are the causes of increased CO2? Either there is increase in the production or there is reduced elimination. Increased production, such as people with fever, exercise, shivering, metabolic activities, this will generate more CO2. Or there is limitation of CO2 washing out due to hypoventilation, respiratory depression, airway obstruction like in asthma, respiratory muscle impairments, obstructive diseases, chronic bronchitis. Yeah, so all these could lead to the decreased CO2 elimination. Now we will reach to the last part of today's lecture, which is about number of disorders: asthma, emphysema, chronic bronchitis. So asthma is the thickening of the airways by the terminal bronchioles, which will be, these usually terminal bronchioles have smooth muscles that will be constricted as an allergic reaction. So it is also going to be inflamed with mucus that is might close the opening of the terminal bronchioles. So this bronchospasm causes the difficulty in breathing and causes hypoxia. In emphysema, there will be activation of certain enzymes that will break down the alveoli, okay, causing hypoxia. Chronic bronchitis is something similar to asthma, but it is not allergic. It's mainly due to smoking. Emphysema is also due to smoking. So this inflammation can cause spasm of the lower respiratory tract, making it very difficult for air to pass to the alveoli. So emphysema is mainly due to activation of the enzymes, as we mentioned. Usually these are inhibited by the anti-protease enzyme, which we have alpha-1 antitrypsin. So normally we don't have this emphysematous changes, but in smoking, the enzymes will be activated, and there are some genetic problems that can also cause emphysema, which will lead to the destruction of the alveoli. Chronic bronchitis, inflammation, edema, thickening of the mucosa, hypertrophy of the muscle with some constriction. Yeah, these are all diseases. Bronchitis, asthma, emphysema, these are all disorders. Yes, emphysema could be genetic and it could be caused by smoking. As you can see here, tobacco smoke, air pollution can lead to bronchial irritation with inflammation, chronic bronchitis, or breakdown of the elastin, the connective tissue of the lungs, causing emphysema. So this will lead to airway obstruction, dyspnea, hypoxia, and it will make the person more easy to develop lung infection. Similar thing also happens in coronavirus infection. The infection with secondary bacteria is more likely to happen. Dyspnea is shortness of breath, difficulty in breathing. Bronchial asthma, lastly. These will be used most likely in the scenarios. So most likely we're not going to ask you about the disease, but the scenario is related to them. So it is good to understand what are these diseases and what happened. When you know these diseases, then you will understand the question better, and you will know how to choose the right answer. So here in bronchial asthma, for example, there will be narrowing of the terminal bronchioles that will make the air more difficult for the air to reach to the alveoli. So nothing wrong with the alveoli. One of you asked, do we have a problem in the compliance? No. Respiratory membrane is normal, compliance is normal, but the problem are in the bronchioles, which are inflamed and narrowed with the hypertrophy and constriction of the smooth muscle in response to certain stimuli such as cold, exercise, smoke, or allergen, and they will make it very difficult to breathe and might cause hypoxia and loss of consciousness in acute attacks. So the drug they take through inhalation will enhance the dilation of the smooth muscle, relaxation, and it will make breathing better. Pneumothorax is another disorder, especially caused by penetration of the thoracic cavity by a knife or any sharp object. I'm sure you are aware about this in movies. For example, those what is the question? Doctor, about the quiz, we cannot go back to the previous question? Yes. This option is not possible. You will be exposed to the question only one time and within a very limited time. So you need to understand the situation right now. So that's the only way it is possible, and it's going to be practiced in all questions. This is so that you, as a student, you cannot go back and change the answer. So the time is limited, and it is not possible to go back and change answers. Okay, so pneumothorax, the last slide. The last slide of pneumothorax. This problem, as we mentioned, explained in movies. Certain people, have you seen them when they are unable to breathe? and then suddenly somebody will use his pen or a knife to stab the chest, and then suddenly blood will come out and the person becomes normal back again and takes a breath. That's pneumothorax. So in pneumothorax, what happens, there is an injury to the chest wall, usually in car accident, causing the air to be trapped inside the lung, squeezing the lung and making it impossible to expand. So it is one sudden change in lung compliance. It is impossible to expand the chest in tension pneumothorax. And the only way to solve this problem is to insert something and open the space and make that air which is trapped inside to move outside from the lung and making it possible for the lung to expand again. So that's pneumothorax. Pneumo means air, thorax, the chest, air in the thorax. So with that, thank you very much. That's the end of the slide. So I will look back into your question. This actually, the process of preventing backtracing is one of the steps which are taken in order to limit the time and limit the any chances that, because you know you are answering from home and it is somehow something to limit changing questions after answering them. You are not used to it, I know, but these measures have been instructed to be followed. Basically, then, once the question is in front of you, you have very limited time, 1 minute. You answer it and you move to the next question. You cannot go back. So you answer that question and that's it, within that very short time. It is no longer possible if we make it in that way. Shunt. What is the meaning of shunt? Shunt is certain amount of blood supposed to go through gas exchange, okay, but it moves from an area with poor oxygenation into an area with poor oxygenation without going for gas exchange. So for example, in the heart, there is some venous blood which is mixed with the arterial blood inside the heart. And in the pulmonary circulation, I think yes, this part is included. You can ask Dr. Hala. Yeah, respiratory system was there. There are questions are there all about the respiratory system. No, answer the question within that time. So you have, let's say, 15 questions, and the total time is 20 minutes. So try to think about each question individually, and then you do your final option, and that's it. You cannot go back and change it anymore. Once you move to the next question, you cannot go back anymore, and that is final. All questions are MCQs, yes. We don't know until now whether the same practice is going to be used for the final. We are waiting the university decision in this regard. But until now, we will proceed with all the quizzes and midterm exams. I don't know, take-home exam. Maybe it refers to open book exam. Certain courses are possible to be done in that way, in which the questions require high order of thinking. It needs your understanding and critical thinking in order to answer that question. This is not possible for anatomy and physiology because most of the information is memorization. So it's not possible because you don't need to think about any difficult situation. If the slides are in front of you, then you can easily answer the questions. That's the problem. So we need to limit the time, limit the back tracing in order to limit the chances. We have been instructed to stop the attendance. There will be no attendance today. So no more attendance will be taken. So with that, yes, lockdown browser will be used. All right. So thank you very much. Wish you all the best. Wassalamu alaikum wa rahmatullahi wa barakatuh. I will stop the